Dear colleagues, my name is Anastasia Ilina. I am a research assistant of the Laboratory of Molecular Mechanisms of Aging at the St. Petersburg Institute of Bioregulation and Gerontology. I am a PhD student of the Department of General Pathology and Pathological Physiology at the St. Petersburg Institute of Experimental Medicine. Today I'd like to focus on neuroprotective effects of short peptides in Alzheimer's disease in vitro. First of all, I'd like to begin with determination of Alzheimer's disease. It's the most common neurodegenerative disease that causes progressive memory loss in older people. Furthermore, Alzheimer's disease is a global medical, social, and economic problem. This is due to the fact that profound memory loss and associated affective symptoms such as apathy, depression, hallucinations, and so on lead to dependence of patients from others in completing of daily life activities. Cognitive, behavioral, and psychological symptoms causes progressive disability of patients and reduced quality of life in AD. Alzheimer's disease is one of the most common reasons of death, occurring from 5 to 12 years after firstly detected symptoms. Speaking on the pathogenesis of Alzheimer's disease, it should be noted that amyloid play a crucial role in AD development. Amyloid peptide is a part of amyloid precursor protein in cell membrane that can be removed by beta and gamma secretase enzymes and released from the cell in extracellular space. Amyloid accumulates in form of oligomer aggregates and amyloid plaques. It should be noted that mutations in APP, PSEN1 and PSEN2 genes contribute to AD development and amyloid plaque formations. Amyloid plaques represent as large round blotches in the histological brain samples of AD patients. Certainly, in control case, we cannot see these accumulations. And what about therapy of Alzheimer's disease? It should be noted that there are 5 FDA-approved drugs for the treatment of Alzheimer's disease. These include 4 inhibitors of acetylcholine esterase that protect cholinergic neurotransmission in neurons. Tocrin, donepezil, rivastigmine, and galantamine are these inhibitors. And last one is memantine, is inhibitor of NMDA receptor. Memantine protects neuron from hyperexcitability. It should be noted that these drugs don't prevent or slow down the neurodegenerative processes. It should be noted that these drugs have considerable side effects. By this reason, development of safe and effective therapeutic agents for Alzheimer's disease treatment is becoming increasingly relevant purpose of modern medicine. Short peptides are promising compounds for AD treatment in view of low immunogenicity, lack of side effects, and high physiological activity. It should be noted that EDR and KIT peptides are active components of dietary supplements Pinealon and Vesugen respectively. Speaking on neuroprotective effects of short peptides, it should be noted the synaptic dysfunctions underlies the cognitive impairments in Alzheimer's disease. Amyloid accumulation contributes to synaptic dysfunction. And what is synaptic dysfunction? Synapse is a contact between neurons that provide neurotransmission, that's conducting of nerve impulses. Dysfunction of neurotransmission consists changing in functional state of synapses that include abnormal formation of synapses and loss of synapse stability. This event triggers decreased number of postsynaptic contacts named as dendritic spines, and changing in the dendritic spine morphology. These consequently events provide learning and memory dysfunctions in Alzheimer's disease. And today we are talking about neuroprotective effects of short peptides, EDR, and KID in Alzheimer's disease in vitro. Let's speak about methods of the present investigation. It was necessary to extract whole hippocampus from the brain of newborn wild-type mice. Next, it was necessary to prepare a primary culture of hippocampal neurons. The experimental groups were following. The first was neuronal culture with applied of anything compounds. The second group was neuronal culture with applied of amyloid beta peptides for development of synaptotoxicity conditions. Third group was neuronal culture with applying of amyloid beta peptides and EDR short peptides as promising neuroprotectors. And the fourth group was neuronal culture with applying of amyloid beta peptides and cat peptides. Next, it was necessary to receive microphotographs of neuronal dendrites by means of confocal microscopy. And on the next step, it was necessary to carry out a morphological analysis of dendritic spines in specific software. It should be noted that there are three main types of dendritic spines that include thin, stubby, and mushroom spines. Research showed that cognitive impairments are closely related with mushroom spines eliminations. And mushroom spines is the most functionally active type of spines. And next, let's speak about the result of present investigation. In normal case, we can see a lot of dendritic spines signed by a white arrow. On the graph, this fact is reflected as a high level of mushroom spines number in control case. In neuronal culture applied by amyloid beta peptide, we can see the loss of mushroom spines number in ED model. In neuronal culture with applying of EDR peptide, we can see a lot of dendritic spines that reflected on the graph as rising number of mushroom spines. It should be noted the statistically significant rising number of mushroom spines. And the last one, in neuronal culture with applied of KID peptides, we can also see a lot of dendritic spines and mushroom spines too. According to these findings, we can suggest that short peptides, EDR and KID have neuroprotective effects in Alzheimer's disease model in vitro, and these peptides improve functional state of neuronal networks in Alzheimer's disease. These facts are very important for future investigations into influence of this peptide on cognition in Alzheimer's disease. In conclusion, we can suggest that EDR and KID peptides as active compounds may be recommended to future investigation in order to develop a safe and effective therapeutic agents for the treatment of Alzheimer's disease. Furthermore, pinolone and mesogan with subtherapeutic concentration of EDR and KID peptides respectively are appropriate to be used in order to prevent age-associated neurodegenerative disease, including AD. That's all I have to discuss in the present lecture, so thank you for your attention.